Good morning, folks. Hey, guys, hear me okay? Good morning, welcome back. How'd you guys like the first exam, lab exam? Easy, right? Now, what I can tell you guys is that you guys need to pay a lot more attention on instructions, reading the instructions. A lot of you folks just put stuff everywhere. Not that you got points wrong for putting them in the wrong order, but it just makes my job harder because I have to grade them manually. There's still a couple more exams that I'm grading. I'm almost done with them, and you should have all of them back today. Okay. Any questions, guys? Anything? Any concerns, questions, problems? Now, the good news is that the hardest exam passed. The first exam, the first lab exam, was definitely the hardest one. Okay. The other two exams, remember, we don't have a final. They will be definitely easier. That does not mean that you don't need to be prepared for the next two exams, right? And they're going to come very, very fast. So we're going to have 3 labs. We have a lab today. Then we have 2 labs next week, Tuesday and Thursday. And then the week after that, on Tuesday, we're going to have our second exam already. So it will come very, very fast. And you guys already have the information to study and prepare for the exam. Don't wait, okay? All right, guys, so we're gonna start the material today for the second exam. And today we're gonna go over the nervous system and the special senses. Also, the good news is that we already covered them in the lecture. So a lot of the stuff that I'm gonna go through is gonna be a review for you folks. And then we're gonna do a lab activity today, which is gonna be watching the Edpuzzle videos. I sent you an email a few minutes ago with the 3 links for the videos you're going to be watching today. And those videos, like the previous videos, they will be videos related to models that I will be using for the exam. So now you understand what I mean by that. You know, I will be using them for the exam, for the next exam. It's going to be a model of the brain, pointing out different structures of the brain. Is going to be a model of the eye, which is going to be on the second exam, and it's going to be a model of the ears. Okay, so very, very important videos to watch. And then, as usual, after you're done with the videos, you're going to jump on Lima and you're going to take 2 quizzes today. So quizzes 7 and 8, and they are always the last thing that we do for the class. Okay? All right, so let me share my PowerPoint with you. All right, folks, so we're going to start with the nervous system, and then we're going to go over the special senses focused on the eyes and the ears, okay? For the nervous system, we're going to just give an overview look at the nervous system. What is the pathway of the nervous system? We're going to spend a little bit of time talking about the neuron and action potential, again, even though you guys are not responsible for this for the lab exam. Then we're going to make a differentiation between the central nervous system, the peripheral nervous system, and talk a little bit about reflexes. And then I'm going to walk through a sheep brain, which is the model that we would, if we were in the lab today, face to face, all of you folks would be working with a brain from a sheep. And there's a lot of similarities between the sheep brain and the human brain in terms of the structure, not so much of the orientation, but in terms of the structure, and we're going to be taking a closer look at those, okay? So talking about the overall pathway of the nervous system, as I mentioned before, our nervous system starts with what we call the receptors, and those receptors, they receive a different type of information. be it sound information, light information, smell information, touch, temperature, etc. Okay, and those receptors basically they're going to sense our environment, both the external environment and the internal environment. Now, the job of the receptors is just that, just to receive that information. And then it's going to pass that information all the way to the brain. And that information is going to travel from the receptors to the brain through what we call the sensory neurons in a pathway that we call the afferent pathway. Okay? So the brain is going to make sense of that information and is going to make a determination how to act, what it needs to be done, if anything at all. Now, the brain itself doesn't do the work. It's just the boss. It just sits up there, okay? And then depending on the response to the stimulus, then the brain is going to send that information all the way down to what we call the effectors. And are the effectors that actually do the work. And an example of effectors include our skeletal muscles and different glands and organs in our body, okay? The information from the brain to the effectors go through a different set of neurons called the motor neurons and through a different pathway that we call the afferent pathway. Taking a closer look at a neuron, and a neuron is quite a big cell in relation to other cells that we find in the nervous system, what we call the microglial cells. And you can see that the neuron has this very peculiar shape to it. It has little finger-like structures coming off from the central part called the cell body. Within the cell body, this is where we're going to find the nucleus. This is where we're going to find different organelles. And then the finger-like structures are what we call the dendrites. Okay. Now, on the opposite side of the dendrites, also attached to the cell body, we're going to have what is called the axon. And you can see that the axon is much thicker than the dendrites, right? The way that the information flow through a neuron is that the information, and by information, it means action potential. So the way that the action potential flows is going to be arriving at the dendrites, passing through the cell body, continuing through the axon until it gets to the axon terminals. Okay? So here we are looking at the flow, the transfer of electrical signals all the way from the receptors and then passing through different types of neurons. And so again, traveling from the receptors to sensory interneurons that are found inside the spinal cord, and then going through the motor neurons and the effectors, okay? Now, depending on the information, some information that's going to travel all the way up to the brain, so the brain can assess that information and respond to that information, or sometimes we have what we call reflexes. And in reflexes, the information do not go all the way up to the brain. It goes straight from the sensory neuron to the interneuron and motor neuron, or it goes straight from the sensory neuron to the motor neurons in what we call true arc or true neuron reflex. Okay. Now we have seen and talked about this before until we turned blue. And here we're looking at a cell that is at rest. And we know that when a cell is at rest, inside is negative, outside is positive. And this resting state is created by the sodium-potassium pump, okay? And that resting membrane potential of a neuron, it's about minus 70 millivolts, while the resting potential of a muscle cell is about minus 85 millivolts, slightly different. And we also understand that the sodium-potassium pump, what it's creating is this huge concentration of sodium outside of the cell, what we call the chemiosmotic gradient. And that as the sodium passes through the ion channels, then we're going to have this change in the polarity inside of the cell from negative to positive, and outside of the cell from positive to negative. And that creates what we call action potential. And action potential is nothing but a what? It's nothing but an electrical impulse, okay? And we understand by now that this electrical impulse and the chemicals, the neurotransmitters that are traveling in the synapses, communicating between neurons, this is what a thought is. When we think about moving a muscle, when we think about anything for that matter, not just moving a muscle, okay, but any thought that we have, they're nothing but chemicals and electricity running through the neurons of our brain, okay? Now, as I mentioned, this action potential is unidirectional, meaning that it only travels in one direction. It goes from the dendrites, cell body, axon, and then axon terminal. Okay. And what we're looking at right here, we're looking at a little slice of the axon, and we can see that this direction of the action potential happens due to what we call local depolarization and repolarization of the membrane. What do we mean by that? When the cell is at rest, like what we're seeing here in this first scenario, how do I know this cell is at rest? Well, I know that because inside is negative, outside is positive. And a cell that is at rest is a cell that is said to be polarized. And this is an important concept to understand, not only now, but to carry on with you, okay? Because this change in membrane potential is a very important concept in biology, okay? And what does it mean by being polarized? Being polarized, and I used this example before in class, being polarized is like having a battery in your hand. Okay, if you have a fresh battery in your hand, that battery is polarized. There is what we call energy potential in the battery. Now, when the battery is in your hand, that energy is not being realized. What do I mean by that? It means that there's nothing happening. There is energy, but the energy is not being used, not being utilized. This is what being polarized means. Okay, now if I take the battery and I put into a device, an electrical device, now the energy potential that is stored in the battery will be realized. You know, the little electrons are going to travel from the negative end to the positive end, and that is going to allow the device to be energized and functional. Okay, so basically this is what polarized means. So when the cell is at rest, there is potential, but that potential is not realized yet. It's not until there is a stimulus to the cell, and that allows the sodium channels to open up, and now the sodium goes inside of the cell, and we can see right here, only in this little area, that we're going to have the action potential being created by the entrance of the sodium. And this is what we mean by local depolarization of the membrane. Now, immediately after this local depolarization, we're going to have the local repolarization of the membrane, meaning that as soon as the sodium channels open and the sodium goes inside of the cell, then those sodium channels are going to close again. And now the potassium channels are going to open, sending potassium outside of the cell, restoring what? Restoring the resting membrane potential. So this is what we call local repolarization of the membrane. So the membrane then constantly do that cycle of depolarization, repolarization, depolarization, and repolarization. Okay. I also mentioned to you the action potential is an all or none principle. And that means that the stimulus to the cell needs to be strong enough to jump over a certain threshold of voltage inside of the cell. And this is the threshold right here. Okay, so inside of the cell, it needs to have enough sodium going inside of the cell to change inside of the cell from minus 70 millivolts to minus 55 millivolts. And when that threshold is achieved, then the action potential is going to go all the way up. Okay, that means that the rest of the sodium channels are going to open, allowing all the sodium to flood inside of the cell. Now, the opposite is true. If this threshold is not achieved, if the stimulus is not strong enough to send enough sodium inside of the cell to jump through this threshold, then action potential will not happen. Basically, the cell is just going to go back to the resting membrane potential. Okay. Right, so let's look at the anatomy of the central nervous system. And folks, if you're sleeping up to this point, it's time to wake up because you know that the lab exams are all about anatomy. And several of the structures I'm going to be discussing right now, you will come across them on the second exam. Okay? All right. So here we have a cartoonish representation of the brain, in which we are looking at the brain from a lateral view. So this side of the brain here would be anterior. This here is posterior. This is superior. This is inferior, okay? Now, when we look at the brain has this very peculiar shape and texture to it. The brain is not a smooth surface. It has these folds and these undulations. So when we look at the external aspect of the brain, we are looking at what we call the cerebrum. Okay, so the surface right here that we're looking at, this is the cerebrum. And we're going to see that we have two what we call cerebral hemispheres. We have a right cerebral hemisphere and we have a left cerebral hemisphere. Okay, and those two hemispheres, they are mostly separated up to a point, and we're going to discuss what is the structure that connects the two hemispheres together. All right, and they function, a lot of it functions separately, the two hemispheres of the brain. Now, when we open up a brain and we look on the inside, we're going to find several important structures as well. One of those structures is what we call collectively the diencephalon. And the diencephalon, which is shown here in purple, okay? So we're going to see that this diencephalon is a group of structures that come together. So we're going to see that there's actually three regions within what we call the diencephalon of the brain. So the diencephalon is not a structure on its own. It's a region of the brain that includes three other structures. Okay. Then here in green, we have what is called the brain stem. And we're going to see that the brain stem as well is subdivided into three subregions. Okay. Finally, all the way in the back of the brain, we have what is called the cerebellum. And the cerebellum is not part of the cerebrum. It is a structure all on its own. All right, so let's go back and talk about the meninges. And this is something, again, we have talked about before in different classes. And we know that the meninges are three different membranes that covers our brain, that helps to protect our brain. Okay, we have the outermost membrane or the outermost meninge, which is what we call the dura mater. We have the middle meninge, which is the arachnoid matter. And we have the innermost meninge, which is what we call the pia matter. And the pia matter is actually touching the brain tissue itself. It's right here. Okay, is the membrane that is actually touching, hugging the brain. Now, in between these membranes, these meninges, we're going to find a space, a very tiny little space, but there is a space between them. For example, there's a space between the dura and the arachnoid, and we call that space the subdural space. Sub means underneath, So this is the space underneath the dural meninge. And then we have a subarachnoid space, which is the space underneath the arachnoid membrane. And both of the spaces here, they are filled with cerebral spinal fluid. Okay? So let's go back to the cerebrum and let's see what the cerebrum is made of. What are the parts of the cerebrum, the structure of the cerebrum? And as I mentioned, the cerebrum is undulated. And what we have done here, and I want you guys to understand what you're looking at. So we're just going to take a little slice of the cerebrum and take a closer look at it. And this is what we would see. Okay, so we're looking at a little section, a little piece of the cerebrum. And we can see how the cerebrum is made up of these undulations right here. Okay. Each one of these undulations, folks, this is what we call a gyrus. When we have more than one gyrus, now we have a gyri. So we can see 3 gyri right here. Okay, but a single one of them is a gyrus. And these little undulations, they are separated from one another by 2 different indentations. Okay, we have a more shallow indentation between 2 gyri, and the shallow one is what we call a sulci. Sulci is plural, sulcus is singular. And then we have a deeper space between 2 gyri, and the deeper one is what we call a fissure. Okay, so again, we can see a sulcus in here, which is shallow, and we can see right here a deeper one, which is a fissure. Okay, now there's a couple of sulci and a fissure that I do want you guys to know by name. 2 of those sulci that are important are what we call the central sulcus, and we have the lateral sulcus. Okay, so the central sulcus is right over here. So the central sulcus actually separates what we call the frontal lobe of the brain from the parietal lobe of the brain. So right here, there's a sulcus right there. That's the central sulcus. And we can see that the central sulcus is also very important because it separates 2 very important regions of the brain that helps to either control our motor functions or control our what we call somatosensory functions. Okay, and again, that is the central sulcus. And then over here we have what is called the lateral sulcus, and this lateral sulcus separates the frontal and the parietal from what we call the temporal lobe of the brain. Okay, now keeping in mind there's different lobes of the brain, the frontal, the parietal, the temporal, the occipital. All of these are bilateral structures, including the cerebellum as well. All of them have 2 sides. You have the right parietal, the left parietal. You have the right frontal, the left frontal, the right occipital, the left occipital, the right cerebrum, cerebellum, the left cerebellum. Okay, and you know by now how important it is to know right and left. Okay. Any questions, guys? Now we have, like I told you, we have 2 cerebrum, right? 2 cerebral hemispheres, the right and the left. And in between, separating the 2 cerebral hemispheres, we have a deep fissure that goes all the way like this, folks, separating right and left. And this fissure is what we call the longitudinal fissure of the brain. Okay. All right, so let's look inside of the brain, and let's just recap here. So the structure that we're looking at right here. Can anybody tell me what is this? What do we call this?
The cerebrum.
Can you be even more specific than that? Which one are you looking at right here? Right or left?
The left cerebrum.
Absolutely. So this is the left cerebrum, okay? Very good. So like I said, the two cerebral hemispheres, they are separated up to a point. So I could actually, literally, folks, take my finger and stick my finger right in the longitudinal fissure. That's my finger right there. I could take my finger and stick it right in between the two cerebral hemispheres in the longitudinal fissure, and I will wiggle and separate those two hemispheres without causing any actual damage, okay, to the brain. Until what point? Until my finger gets to this structure right here. And this structure is what we call the corpus callosum, okay? And this corpus callosum here is basically the region of the brain, the structure of the brain that connects the right side of the brain with the left side of the brain. So there's going to be a lot of nerve endings that is going to be running from right to left, sending information from the right cerebrum to the left cerebrum, okay? Now let's look at the diencephalon. And as I mentioned earlier, the diencephalon is subdivided into 3 regions. And let me show you here where the diencephalon is. So this whole region here, this is the diencephalon. And the diencephalon is made of what we call the thalamus, right here in the center. We have the hypothalamus right over here. And hanging from the hypothalamus, which is part of the hypothalamus, we have a very important gland, which is the pituitary gland, okay? Does anybody remember what is the name of the bone marking and the bone in which the pituitary gland sits? You guys were asked that yesterday to identify the structure. So what is the name of the bone marking and the bone where the pituitary gland is going to be sitting right inside? You guys are like, oh, I already done that. I don't need to remember that anymore. No, you got to carry that information with you. Come on, anybody.
Is it the sella turcia of the sphenoid bone?
Yes, that's the sella turcica of the sphenoid bone. The sella turcica of the sphenoid bone, okay? Very good. And the third part of the diencephalon, so again, we have, the thalamus, the hypothalamus. The third part is what we call the epithalamus. And within the epithalamus, we find a little gland right here. And this gland is what we call the pineal gland, which is responsible for producing melatonin, okay? Down here, right inferior to the diencephalon, we're going to find the brain stem. And the brain stem is our most primordial brain, folks. The most basic information, functions that we have, such as what? Such as the beating of our heart, such as our breathing, all of those functions, they are controlled, they are regulated by regions of the brainstem. When we evolved during the process of evolution of who we are today, we started with the brainstem. That's all we had. Okay, the early forms of what we evolved, the animal that we evolved from, that's all they had, just a brainstem to maintain its most basic functions. Then from the brainstem, we evolved into the other parts of our brain, okay? And the brainstem is subdivided into what we call the midbrain, the pons, and the medulla oblongata. Beyond the medulla oblongata, now we get into the spinal cord. right there. And then the other parts right here that we can see also the left cerebellum, and we can also see this white structure right here, and that is what we call the arbor vitae, or the tree of life of the cerebellum. We don't fully understand what is the function of the arbor vitae, but we know that the overall function of the cerebellum is to help with our motor coordination and balance. Taking a close look at the spinal fluid and where the spinal fluid is located. Not only we find the spinal fluid in those subdural and subarachnoid spaces, the spaces between the meninges, but we also find the cerebrospinal fluid inside of the brain. So inside of the brain, we have these little channels, these little caves in which cerebrospinal fluid is filling inside and circulates inside. Okay. So let's look at those spaces. There's two big spaces right here. Let me circle them for you. So this here is what we call the lateral ventricles. Okay. So the lateral ventricles here, we do have two of them, the first and the second, but they're collectively what we call the lateral ventricles. And the lateral ventricles are positioned right here. Let me draw this better. These things look like a horn right here. So these are the lateral ventricles. Then from the lateral ventricles, the cerebrospinal fluid is going to flow into what we call a third ventricle, which is positioned about here and here. Then this third ventricle is connected to the fourth ventricle through a little channel, a little tube that is called the cerebral aqueduct. So this little region here is the cerebral aqueduct. And then which is going to connect the third ventricle with what we call the fourth ventricle, which is positioned right over here, right on the base of the cerebellum. Okay. After the fourth ventricle, now the cerebrospinal fluid is going to get inside the central canal of the spinal cord. Okay, so again, the cerebrospinal fluid is going to keep floating from one place to another. Very briefly here, let's look at now at the peripheral nervous system. And the peripheral nervous system is made up of two groups of nerves. We have what is called the cranial nerves, and we have what is called the spinal nerves. What is the difference between the two? The cranial nerves are a set of 12 nerves, and we can see them right here. And these nerves, they are coming off, they are branching off straight from the brain. This is why we call them the cranial nerves. And most of these nerves here, but one of them, they will stay within the head region and the neck region. Does anybody remember what is the only nerve that will leave the head and the neck region and innervate the thoracic and the abdominal region?
Cranial nerves.
The vagus, this guy right here. So the vagus nerve is the only nerve out of the 12 that will leave the head and the neck region and innervate the thoracic and the abdominal region. In fact, this was one of the questions on yesterday's exam. And I also asked you guys about this one right here, the vestibulocochlear nerve. And I told you guys to remember those two in class, okay? If you're paying closer attention, you would have known the answer to those questions. And this vestibulocochlear nerve, we're going to take a closer look at it because this is the nerve that branches off out of the ears, both the cochlea and the vestibule, and is sending sound or hearing and balance information to the brain. Okay? Now, the spinal nerves, these are nerves that branch off the spinal cord, and there are several of those. Now, you guys are not responsible for this in the lab exam. I will not ask you specific questions to name the spinal nerves. But we do understand, and this is something I mentioned before, that different parts of the spinal cord, they have different sections. For example, we have the cervical section, or the cervical nerves, the thoracic nerves, the lumbar nerve, sacral, and coccygeal nerve. Just like we have the cervical curvature, we have the thoracic curvature, and so forth. A couple of nerves here that I could ask you guys on the exam, and I color-coded them for you. This is something you might come across on our second exam. One of those nerves are what I call the radial nerve. And blockage to this radial nerve can cause what we call the wrist drop. Another one is an ulnar nerve that is running right next to the ulna, and a blockage or damage to this nerve could cause what we call the ulnar nerve palsy. Another possible nerve I could ask you guys is the medium nerve. And this medium nerve damage or blockage could lead to what we call medium nerve palsy. A couple of important nerves in our legs, and there are two of them only that I want you guys to know by name that I could ask you on the exam. And actually, there's three of them. We're looking at two of them here in the anterior BIOL, and we're going to see another one in the posterior BIOL. And those are the femoral nerve and the obturator nerve. Remember that this obturator nerve, this thick nerve right here, is going to pass right through the obturator foramen of the hip bone. Okay. And in the posterior BIOL, without a question, the most important one is this guy right here, the sciatic nerve. And you can't miss it because it's a very thick nerve that goes all the way from the hip region and basically extends down the whole thigh before it changes name and now becomes what we call the common fibular nerve and the tibial nerve. And the tibial nerve will continue down all the way to the foot. Okay, so these two here, the common fibular and the tibial, they are branches of the sciatic nerve. Moving on, very quickly here, I want to talk about reflexes. And we understand that reflexes are very fast. Reflexes are very quick. Now, not all reflexes are created the same. We have what we call somatic reflexes and autonomic reflexes. Can anybody tell me what is the difference between the two? We have already seen this before. What is a somatic reflex? What is an autonomic reflex?
Somatic reflexes are more on the skeletal muscles, and then automatic reflexes are like stuff that we can't control, like the blinking, like the how our heart pumps and stuff.
Yes, and it's not automatic, autonomic reflex. And yes, so somatic reflexes are the ones that control our skeletal muscle or stimulate the skeletal muscle. Okay, such as the patellar reflex, such as the Achilles tendon reflex, and then autonomic reflexes are the one that control that stimulates either our cardiac muscle or the smooth muscles or glands, which are not under our direct control. Okay. Now, these reflexes, they can either be what we call a 3-neuron reflex arc, or they can be a 2-neuron reflex arc. What is the difference between them? And by the way, the 3-neuron and the 2-neuron reflex arc, they belong to the somatic reflexes, okay? They are part of the somatic reflex. And what is the difference between the two? The only difference really is the number of neurons that are involved with that reflex. If you have all 3 different types of neurons involved, the sensory, the inter, and the motor, then we have a 3-neuron reflex arc. If we have just 2 neurons, just the sensory and the motor, the inter is not involved, then we have what we call a 2-neuron reflex arc. You know, the patellar reflex, and the patellar reflex is when we hit a little rubber mallet right here, right underneath the patella bone, and then we kick our leg forward. That is an example of a 2-neuron reflex arc. Okay. All right, and the last thing I want to talk about this lab before we move on to lab 8 is that I want to show you some of the structures that we have been talking about, but looking at them using a sheep brain. Okay, not a human brain, but a sheep brain, which is a good model, a model that we use in the lab and other institutions. They use them to learn more about brain tissue. Okay, using a biological sample, not just a model. So here we're looking at a superior view of the sheep brain right here, meaning that we're looking from the top. Okay, and what can we identify right here? So we can identify the different hemispheres. So we have the right cerebral hemisphere. We have the left cerebral hemisphere. And then separating the 2 hemispheres, we have this line right here, right in the middle. And what is this line? This is what we call the longitudinal fissure. Remember that I mentioned to you, we can wiggle it and separate them with our finger until we get to what point? Until we get to this point right here, the structure. And this structure is the corpus callosum. Okay. What else can we see here? Well, we can see the gyri, and remember the gyri are the undulations on the brain. Gyri is plural, gyrus, G-Y-R-U-S is singular, is one of those. We can see the different lobes of the brain. So right here would be the frontal lobe, then we would have the parietal lobe, over here would be the temporal lobe, and then all the way in the back we have the occipital lobe. Be careful not to confuse the occipital lobe of the cerebrum with the cerebellum, okay? Which is a completely different structure from the cerebrum. Right? Over here we have an inferior view of the sheep brain. And we can also identify some structures here that we are unable to see it from the superior view. For example, we can see a couple of the cranial nerves. We can see the olfactory bulbs right here. And these olfactory bulbs, these are the nerves that are responsible for our sense of smell. We can see a little piece, not all of it, because it's dissected, but we can see a little piece of the optic nerve. And the optic nerve are the nerves involved with our sight. We're going to talk more about them in the next lab today. And I mentioned to you folks that the information, like the optic nerves, for example, the information, the nerves, they are crossed, right? When you look at something with your right eye, whatever you're looking at, that information is being processed on the left visual cortex. When you look at something with your left eye, that information is processed in your right visual cortex. Okay? At the point where the 2 optic nerves cross, this little region right here, this is what is called the optic chiasm. So the optic chiasm is nothing but the point where the optic nerves cross each other, okay? Going from right to left and left to right. We can also see part of the diencephalon. So we can see the pituitary gland right here, which hangs from the hypothalamus. And we also can see the brainstem. We can see the pons, and we can see the medulla oblongata right here. The midbrain would be a little bit here in front, okay? And we know that beyond the medulla oblongata, then we have the spinal cord. Now let's do this. Let's cut the brain right through the longitudinal fissure, and then look at this left, look at the left cerebrum here, the left half of the brain. So we can see the cerebrum, which is right over here. Right underneath the cerebrum, we can see the corpus callosum, which is this region right here, in which the right and the left hemisphere connect with one another. Okay? We can also see the lateral ventricles right here. And I want to point something out here that is shown in this picture. Can you see that there is the space here that I just circled for you? And that is the space itself is the lateral ventricle, where we have the cerebrospinal fluid, okay? But then there is this name here, septum pellucidum. What is the septum pellucidum? Septum pellucidum is a little membrane that separates the lateral ventricles, that separates the first ventricle from the left ventricle. So there's very thin membrane that separates the two ventricles. And so if you're very careful when you're dissecting the brain, you can leave intact this membrane. You can see a membrane right here. And this membrane that separates the ventricles, the first from the second, is what we call the septum pellucidum. Okay. We can see the thalamus over here, which is a big part of the diencephalon. We can see the optic chiasm, which is the point where the optic nerves cross each other. We can see, what else? We can see the cerebral aqueduct right over here. And the cerebral aqueduct connects the third ventricle, and the third ventricle would be about here in the shape. So connects the third ventricle with the fourth ventricle. So the fourth ventricle would be all the way back here, right at the base of the cerebellum. We can see the left cerebellum with the arbor vitae, which is the white tree of life structure in the cerebellum. We can see the pons, medulla oblongata, and beyond that, we can see the spinal cord.
Professor, in the picture, the second picture, is the white stuff, is that white matter?
Right here in this picture?
Yes.
What white stuff? This right here?
Yes.
No, this is not the white matter. This is just a membrane that holds the brain together. So this brain has been dissected, right? So the white matter, we don't find it until we cut the brain on the inside, okay? For example, the white matter would be here. When we cut through the corpus callosum, we would see white matter. So the corpus callosum is made up of white matter. Any other questions, guys? All right. So moving on now to lab 8. In lab 8 is going to cover general and special senses, but focus on the eyes and the ear. Okay. Now we have 5 different type of what we call the special senses. And we don't have just the eyesight and the hearing. We also have a touch. We also have a taste. We also have smell. Okay, so touch, smell, taste, hearing, and sight. These are what we call the 5 special senses. The only thing I want you guys to know about the touch at this point is that every single different feeling that we have when we touch something, you know, the feeling of pressure, the feeling of temperature, the feeling of texture, these are all picked up by different type of receptors that we have in the skin. So what I'm saying is that there's no one single receptor in our skin that will perceive all of that information. Okay, every one of those informations, they are picked up, they become activated by different stimuli. Okay, so for example, we have nerve endings just for pain and temperature. We have nerve endings just for texture. We have specific nerve endings just for pressure and weight. All right, moving on here, let's talk about the eyes. And you know, it really is a pity that we are not in the lab face to face, because the eye lab is my favorite of all the labs. And that is because when we do this lab, we would be working with a cow eye. Okay, I would divide you guys into groups. Everybody would have a big old cow eye, which is about the size of a golf ball. And you would be able to cut through it and look at the different structures. Everything that we're going to be discussing and talking about it right now, you would be able to see it, you know, as you go and dissect the cow eye. We're going to watch a little video later today that goes through the dissection of a cow eye and describes some of the structures that we're able to see it. Okay. Inside of the cow eye, there is this membrane right in here, in between what we call the retina, right underneath the retina. There's this membrane that is called the tapetum lucidum. And the tapetum lucidum, and we're going to see this in a minute, but this tapetum lucidum is only found in animals that are active at night, like cattle, for example, or cats and dogs. And this tapetum lucidum, it has this metallic, bluish, greenish coloration to it. And in my opinion, is the most beautiful natural color that I have ever seen anywhere, any place, is this coloration, the color of this tapetum lucidum. And what this tapetum lucidum does is that when the light enters the eye of animals that have the tapetum lucidum, and by the way, we don't have them, we don't have the tapetum lucidum. But when the light enters the eye of animals that have tapetum lucidum, because of this metallic coloration of the tapetum lucidum, the light starts bouncing around inside of the eye. And what that does is that helps you enhance the little amount of light that is coming inside of the eye of this animal, especially at night. This is what gives those animals night vision, and they're able to see a lot better than us at night because of this bouncing around of the light due to the presence of the tapetum lucidum. Okay, and we're going to see the structure in a minute. Now, our eyes are subdivided, are made by 3 layers. We have an outer layer that is called the fibrous layer, and the fibrous layers include the cornea, which is a clear layer in the front of our eye. And we have the white of our eye that we call the sclera. Okay? In the front of the eye, folks, so let's say that, you know, this is the opening of your eye right here. Okay? In the front section of the eye, covering the sclera, we're going to find a clear membrane called the conjunctiva. So don't confuse the conjunctiva with the sclera. So the sclera is the white structure, the white tissue here. The conjunctiva is this thin, translucent, transparent membrane that is covering the sclera in the front of the eye. Okay? When somebody has conjunctivitis or pink eye, it's because there is infection, bacterial infection of the conjunctiva. Okay. The middle layer of the eye is what we call the vascular layer, and that is made up by the choroid, in which we have a lot of blood vessels running through it. It's made up of the ciliary body, also called the ciliary muscle, and made by the iris, which is a smooth muscle that controls the opening of the pupil, the size of the pupil. And the pupil is just this space. Okay? And then the innermost layer is what we call the retina. The retina here is shown in yellow, and it's called the sensory layer because this is where we're going to find the photoreceptors that allow us to perceive light. a couple of the structures that are very important, and you guys will be asked about them in the next exam. One of them is this right here, this nerve, which is one of the 12 cranial nerves. And I need you guys' attention here. Do not confuse the optic nerve with what we call optic disc. The optic disc is right here, folks. Just this little section, this oval section right here. That's the optic disc. The nerve itself that is behind the eye, which is going to continue here. This is just a little cut section. The nerve is the optic nerve. So don't confuse the optic nerve with the optic disc. Two different structures, okay? So the optic disc is also what we call our blind spot. And we're going to see, and I talked about it on in the lecture, but we can do a little test to check for our blind spot. And it's called the blind spot. Why? It's called the blind spot because there are no photoreceptors right here in this region. This is the region that the axons of the photoreceptors in the retina, they are leaving the eyeball. And then they are bundling together to form what? To form the optic nerve. So when the light hits the optic disc right here, we cannot see the light. Okay, this is our blind spot, right? The final structure I want you guys to know here is this little oval region, the little darker region here in the retina, and this is what is called the fovea centralis. What is special about this fovea centralis? The fovea centralis is where we have the highest concentration of photoreceptors. And when the light hits the fovea centralis, this is where we can see the sharpest. So when we look at something straight at it, the light that is bouncing on whatever we are looking at and then coming inside of our eye is hitting the fovea centralis. This is why we can see it the sharpest. Now, does that mean that is the only spot where the light is hitting in the retina? No. The light is also hitting elsewhere. And this is what gives us our what? This is what gives us our peripheral vision. When we stare at something straight at it, I can still see, without looking at it, I can still see what is here to the side of my eye, what we call the peripheral vision. Okay, because there are still photoreceptors there outside of the fovea centralis, but I can't see it as sharp. Now, if I move my eye or my head, and then I look at that, straight at it, now the light that is bouncing on that object is going to reach the fovea centralis. Okay? Any questions, folks?
Can you state again what's in charge of our peripheral vision?
What is in charge of the peripheral vision? What do you mean by it?
Like what part of the eye makes us see like other items in
the part of the eye is the same as the retina. So the difference between when we stare straight at something, okay, and what we look at on the corner of our eye with the peripheral vision is that when we stare straight at something, the light that we are looking at, that object, we're staring at it, is hitting the fovea centralis. Now, at the same time that we have a light bouncing from the object that we're looking at right here in front of us, at the same time that there is light rays that is coming straight through and reaching and hitting the fovea centralis, there is also light hitting elsewhere outside of the fovea centralis on other spots within the retina. And that is what gives us the peripheral vision. The light that is hitting elsewhere but the fovea centralis, still within the retina. Okay. Is that clear? Yes?
Yes. I have one more other question.
Of course.
It's like it's kind of dumb, but like,
It's no
do you
dumb
know,
questions.
do you know why we like blink so much? Like, why can't our eye like withstand like not blinking or something like that?
That's a good question. We blink because when we open our eyes, when our eyes are open, our eyes get dry because of the wind, because of the temperature. So there is moisture in our eye. It's being constantly made by the lacrimal glands. Okay, when we have our eyes open, that moisture in the air and the atmosphere is going to suck the moisture out. And this is where, when we blink, we basically re-wet our eyes. That's the main function of blinking. And also we blink to remove stuff that gets in our eye. It's like a reflex, in fact, the blinking, right? When something gets into our eye, or even if we take our finger and we just, bring closer to our eye, we have this reflex, this automatic reaction in order to protect our eye by blinking, like, closing our eyes. Any other questions, guys? All right. A couple of other structures I want to mention here that you will come across in the exam. Like I said, this region right here, this is the ciliary body. And the ciliary body has another name too. It's also what we call the ciliary muscle. Don't confuse the ciliary body with the iris. Okay, so the iris in the front of the eye here is the colored part of the eye. That's the iris. We cannot see the ciliary body. The ciliary body is inside of the eye. Okay. Also, we have different chambers in here, different spaces. There is this space in the front of the iris right here, in between the cornea and the iris. And this space is what we call the anterior chamber. And this anterior chamber contains aqueous humor right here, which is a fluid, okay, just like water. Now, in the back of the iris, we're going to find the what? We're going to find the posterior chamber. In this posterior chamber, we're going to find not only aqueous humor, the same fluid that is here in the anterior, but we're also going to find what is called the vitreous humor. And this vitreous humor is a much more viscous fluid. It almost looks like jelly, okay? And this vitreous humor is very important because it keeps the shape of the eye. If we lose fluid from the eyeball here, the sclera is going to collapse or is going to create wrinkles. And that could cause wrinkles to the retina, and we're not going to be able to see. Also, the humor inside the posterior chamber keeps the retina attached to the back of the eye. If we lose the fluid, let's say you damage your eye, you have a hole in your eye, and now the fluid is leaking, that could cause the detachment of the retina. And if your retina detaches, basically it's going to peel off. Now you're not going to be able to see. This is an emergency procedure in which you need to go see a doctor to have your retina reattached to the back of the eye. Okay, so these fluids are very important. Also, do not confuse the lens with the cornea. A lot of students do that. Two different structures. Okay. So let's go very briefly here through the different photoreceptors. What are they? What are the different types? We have a group of photoreceptors that are called the rod photoreceptors, or the rod cells. They are cells, okay? And we have a group that are called the cone cells. The rod cells are involved with our dark vision. The cone cells are involved with our color vision. And I'm gonna explain a little bit here about the difference between the two. Among the cone cells, we have three different types of cells. We have the blue cones, green cones, and red cones. Now, it does not mean that these cells are color. So the cells are not blue or green or red. What does it mean then? What it means is that the blue cone cells, they only become activated. And when I say activated, it means what? Well, they become stimulated and they generate action potential. That only happens when they're exposed to light in the range of around 400 nanometers. The light that is, this light here is only going to activate these cells. Then the green cones, this means that these cells here are only going to become activated in light in the range of around 550 nanometers. And then the red cones, these cells are only become activated on light that has a range of around 700 nanometers wavelength. And that what allow us to see different colors. When those different cells are activated by different visible light, then the combination of the activation of the cells then it gives us the different colors of the rainbow. Okay, you might have heard of people that are colorblind. What does that mean? Does that mean that they can't see color at all? No. All that it means is that some folks might have a decreased number of one of these different type of cone cells, which means it might make it more difficult for them to be able to perceive certain colors. Okay. Now, what about the rod cells? What do we mean by dark vision? Look, the rod cells are involved with dark vision because these cells right here, they become activated. And again, when I say activated, means that they generate action potential. They become activated by very little amount of light. Okay, light that will not activate the cone cells yet. Let me give you an example. When we walk into a dark room, now it's not pitch black, it's dark, you can still see around, okay, but it's pretty dark. Do you see any color, or you just see things in shades of gray and black? Do you see any color when you walk into a dark room?
No.
No, you don't. Why not? The answer is because the amount of light that there is in that room is enough to activate the rod cells, but is not enough to activate the cone cells. And that is why we don't see color in the dark. Now, let's say that we have one of those light with a dimmer, okay? And you can slowly increase the intensity of the light in that room. Then slowly, as you increase the amount of light in the room, now you're going to start seeing color. Why? Because now the cone cells are becoming activated. Okay? And rod cells, they need, in order for them to become active, to work, they need a protein that is called rhodopsin. And this rhodopsin protein requires vitamin A in order to be synthesized. So if you don't have vitamin A or enough vitamin A, you cannot make the rhodopsin protein, which means you're going to have a difficult time seeing at night. This is what is called night blindness. Okay. Any questions, guys?
Professor,
Uh-huh.
if someone is like completely blind, does that mean they don't have like any photoreceptors in their eye?
Good question. So there's different reasons why people are blind. The most serious, it could be, for example, the same way that there are different reasons why people are deaf. And it could be a damage in terms of the conduction of the sound, or it could be a nerve problem. Okay, so let's say somebody could be blind. All their photoreceptors are working, okay, but then there's damage to the optic nerve. So the photoreceptors work, the photoreceptors become activated, but then they cannot send the information from the eye to the brain. So it has nothing to do with the photoreceptors per se. Now, it could be that somebody is blind because the photoreceptors are not working. So it really depends. Any other questions, guys? All right. And here's what I was trying to describe to you, which are different wavelengths of light and how the different wavelengths of light is going to activate different cone cells, which is going to give us the perception of different colors. Okay. Now, in this little simulation here, we are comparing a side-by-side comparison of the same VIL of a human eye and a cat eye. Can anybody tell me which one of this two here is the VIL of a cat at night? Is the picture on the top or the one on the bottom?
The bottom.
Absolutely. Okay. And we can see a huge difference here. You know, in a human eye, we're going to be able to see only about where the light is shining right here. Now look at the cat's eye. You're not only going to see the section, basically going to have a much more broad VIL of that scene. Okay. Now, why is that? That is because of the tapetum lucidum. And this here is that little blind spot test that I told you guys about it. And again, this is something that you can do on your own. And it's really kind of a freaky when you are looking at an object, okay? And then you change the distance of the card in front of your eye. And then all of a sudden, you don't see the spot in your peripheral vision. Okay, so again, how is this done? So you're going to have a little index card. You're going to make an X on one end and the dot on the other end. And you're going to close one eye and you're going to stare straight at the X. Now, when we stare at the X, so let's say I have a card right here. My hand is the card, okay? When you stare at the X, I can still see the dot with my peripheral vision, all right? Now, when I bring this index card, it's still staring at the X, and I bring it closer to my eye, there's going to be a distance from my eye in which the dot will disappear. And what's happening? Why does the dot disappear? It disappears because now the light that hits the dot and then bounces back inside of my eye is going to hit the optic disc, the blind spot. And when it hits the blind spot, I can't see that image. Okay. All right, so let's take a closer look at the cow eye dissection. Like I said, this is what we would be seeing and doing in the lab today. And again, we can still able to see a lot of the structures that we saw in looking at the cartoon. You know, we can see the cornea, we can see the lens, we can see the ciliary body. And I don't know how well you guys can see. You're going to see it better when the video that we're going to watch today. By the way, this cream color structure here on the surface, that's the retina. And the retina is very, very thin. Okay. And right underneath the retina, you can see there's a little bluish coloration right here. That's the tapetum lucidum. And that's what allows animals that are more active at night to be able to see better, have a better night vision. All right, moving on. Now I want to talk about different parts of the ear. And you guys are also responsible for these in the next exam. So the brain, the eyes, and the ear is a huge part of the exam, okay? And I mentioned in the lecture that our ear is subdivided into three main parts. We have the external ear, the middle ear, and the inner ear. The external ear, one of the parts of the external ear is what we call the auricle or the pinna. And that is what we call the ear. It's just this piece of cartilage, this tissue right here, okay? What do you guys think is the function of this auricle, the pinna? Is it just to hang earrings from, to look pretty? Why do you think we have them? One of the things I want you guys to understand, and I mentioned this before, everything in our body has a function. If it is there, it's because it has a function, or we are in the process of losing the function and losing the structure. An example is our, tailbone, our coccyx. The tailbone is just a residue of a tail that we used to have, back in evolutionary times. Okay, the more we evolved, it's going to come to a point in which that bone is going to be gone from us completely. Okay, so always think of that. Whatever we are looking at in our body, structures that they have a function. So what do you think is the function of the auricle?
Does it like direct the sound waves into our ear?
Absolutely correct. The auricle, it works like a funnel. Look at the shape of it. Let me draw a funnel right here. So this is what a funnel looks like, right? So the auricle looks and functions just like that. And what the auricle, the pin is going to do is going to take sound waves coming like this. And then they're going to direct those sound waves all the way inside the external acoustic meatus, inside the auditory canal. From the auditory canal, the sound waves then is going to get where? Is going to get right here, the tympanic membrane, also called the eardrum. And then the tympanic membrane, the eardrum, they are going to vibrate, just like my hand here. The vibration then is going to be picked up by the little auditory ossicles, which are tiny little bones inside the middle ear. The first bone here that is directly attached to the tympanic membrane is the malleus, and this guy is going to vibrate. Then the malleus is going to send the vibration to the incus, right here in the middle. Then the incus is going to send the vibration to the stapes, right here. And the stapes is attached to the vestibule, which is the bone of the inner ear. And then the vestibule is going to guide that sound wave, that information, all the way down inside of the cochlea. Okay, basically this is the flow of the sound wave until it gets to the structures inside of the cochlea, inside the ears, that is involved with hearing. All right. Now, I shouldn't have to say this, but keep in mind that we have 2 ears. Our ears are bilateral structures. So each one of the structures right here, the auricle, the external acoustic meatus, the malleus, the incus, the stape, the cochlea, the vestibule, the vestibulocochlear nerve, they are all bilateral structures. And as such, you need to tell me if they are right or left, okay? Now, so let's look at the bones of the inner ear. Like I said, we have the vestibule, and on the top of the vestibule, we have this arch here called the semicircular canals. At the base of the semicircular canals, where they are attached to the vestibule, this region right here, it's called the ampulla. And the ampulla is going to be involved with our equilibrium, okay? As well as the base of the vestibule right here. Then on the opposite end, we have the cochlea. And we're going to see inside the cochlea, this is where we're going to find the apparatus that is involved with our sense of hearing. Right here in the back in yellow, we see 2 different nerves. One is the cochlear nerve, right here, that is branching off the cochlea, bringing sound information to the brain. The other one is the vestibule nerve, also called the vestibular nerve. And this one here is branching off the vestibule. And this one here is responsible for our sense of balance. Okay. The final structure I want to show you here is this tube right here that connects the middle ear with our throat. And this tube is what we call the pharyngotympanic tube, also called the auditory tube or the eustachian tube. Warning, guys. Do not confuse the auditory canal with the auditory tube. They all have the same name here, auditory. One is the canal, which is the external acoustic meatus. The other one is a tube, which is the pharyngotympanic tube. They describe two completely different structures. So let's look at inside the cochlear region of the vestibule. So let's amplify this cochlear region here and open it up. This is what we would see. We see that these bones here, they are not solid. There are spaces inside of the bone. And the spaces here, they are filled with fluid, okay? In one of the spaces, we're gonna find this structure right here. And this is what is called the spiral organ of Corti. Let's amplify the spiral organ of Corti right here. And inside the spiral organ of Corti, we're going to find this membrane here on top, which is a tectorial membrane. And right underneath this membrane, we have little nerve cells that are called the hair cells. They have nothing to do with hair, okay? It's just a name for the cells. These are receptors. These are nerve cells. When the vibration that is coming from the vestibule and then getting all the way in the cochlear region is going to allow for the vibration of the fluid inside of our ear. Basically, they're going to create like little waves inside of our ear. When those waves get all the way here inside the spiral organ of Corti, this tectorial membrane is going to undulate. When they undulate, the little hair cells that are underneath it, they become stimulated. When they become stimulated, they create what? Action potential. And then this action potential is going to travel through the cochlear nerve all the way to the brain, and we're going to be able to, the brain is going to make sense of that information. Okay? Now, I mentioned that we have two different types of deafness. We have either what is called the conduction deafness or sensory neural deafness. And the conduction deafness is when the sound wave is stopped. If you had, let's say, have a big old earwax right here, the sound wave that is traveling through your auditory canal is going to be stopped. You might have a little bit of a signal coming through, but they are significantly dampened, decreased. That is an example of a conduction deafness because you're stopping the propagation of the sound wave. Now, sensory neural deafness, that has to do with damage to either the hair cells inside of the cochlea or damage to the cochlear nerve itself. Okay? It's just like when I mentioned about blindness. It could be blindness because of the photoreceptors, or it could be blindness because of the optic nerve itself.
So when people grow old and they have hearing problems, is it because, which is more likely, is it because of conduction or sensory neural?
Excellent question. When people get older, most of it has to do with sensory neural. That is not necessarily the nerve, but the hair cells. So the hair cells themselves, they can start dying off. And these are nerve cells. And remember, nerve cells, they don't grow back. And once the hair cells start dying off, that stimulation is not going to happen anymore. We're not going to have action potential being generated and send information to the brain. Okay? So most common issue as we get older, just like anywhere else in our body, as we get older, our cells die off, due to regular damage to the cells. And we are lucky in some other parts of our body, other functions, those cells can grow back. But for our ears, the hair cells, they don't grow back. Any other questions, guys?
When do you think that like listening to music in like earphones a lot will damage the hair cells, or what do you think?
100%, yes. And the problem is of both cases, also conduction and sensorineural. One of the problems with listening to loud music with earphones all the time, and also people that work in like construction or, you know, rock bands, is that you can actually cause cracks and damage to the ossicles. The ossicles are like a bone, like any other bone in our body. They can and will break when they receive a lot of stress. So when you're listening to loud music all the time right in your ear, you create a lot of vibration on those bones, and those bones could crack and damage, and that on itself is going to lead to deafness. But also the loud sounds can also damage the hair cells as well. Any other questions, guys?
Professor, do you like know what happens when our ears get like plugged?
Sorry, say again, Ayana?
Do you know what happens like when our ears get plugged? Is that like conduction deafness?
Get clogged. It might be due to, what do you mean by clogged? There's a couple of
like
reasons
you're
for
in
it.
an airplane and then
Okay,
ears, like,
it's actually a good question. You know, the main function of the pharyngotympanic tube right here is to help balance and equilibrate the pressure in our ears. So by having this connection between the middle ear and the throat, we can help again to balance out and equilibrate the pressure inside. So when we change altitude very fast, like in going in an airplane or diving, under the water, what's going to happen is that the pressure in the air causes an increase in the pressure in the middle ear, which we are unable to fully equilibrate using the pharyngotympanic membrane. And that's what caused that clogged feeling that we have. Okay. Now that could also be to something else. The clogged feeling, it could be due to a middle ear infection. And in a middle ear infection, we can have accumulation of fluid. Now there is naturally fluid inside the middle ear. Okay, our whole ear is filled with fluid. Okay. Now that the amount of fluid could increase if we have a middle ear infection. And that on itself could give us that feeling of cloggedness and fullness in our ears. Good question, guys. Any other questions? All right. Very briefly here, I want to talk about equilibrium, which is another important part of our function of our ears. And we do have two different types of equilibrium. We have static or dynamic. Static is just when we stand still. Dynamic is when we spin around. And those two different equilibriums, they are going to be sensed by different parts of the ear. The static equilibrium is sensed right here at the beginning of the vestibule, this region right there, okay? In which inside, we're going to find space filled with fluid. Floating on top of the fluid, we have tiny little stones of calcium called the otolith, and embedded, swimming, submerged in the fluid, we have the little hair cells, which are the nerve cells. The same, by the way, hair cells that we have in the cochlea, okay? Now, when we move our head forward, gravity is going to pull on the otolith, pull on the fluid, and that also is going to pull on the little hair cells, and they physically move forward. When they move forward, that's going to send information to our brain, telling our brain the position of our head. We don't need to have our head open, our eyes open, to know what is the position of our head, okay? Simply due to the movement and the positioning of the little hair cells in the vestibule. Now, dynamic equilibrium that is perceived at a different spot. They're perceived right here at a region that we call the ampulla. which is right in between the vestibule and the semicircular canals. Okay. And again, inside of the ampulla, there's a space filled with fluid. There's always fluid in the ear. And in the ampulla here, we have this little membrane called the cupula, which is very similar to the pectoral membrane inside of the spiral organ of Corti. And this cupula, when we spin around, the cupula will also spin in the same direction that we are spinning. And embedded in the cupula, we have, again, hair cells right here. And once those hair cells, they move, they become activated, they generate action potential, and send that action potential to the vestibular nerve. Again, sending information to our brain on the position of our head. Okay. All right. I'm going to show you this video here. So this is not the activity we're going to be doing today. Sorry. The activity today is going to be the head puzzle. But before we get there, I want us to watch this little video here. We're not going to watch all of it.
Today we're doing the cow eye dissection, and let's begin by looking at some of the external anatomy of the cow eye. We're going to start with the outermost structure right here, the most anterior structure, and this is the cornea. In an actual cow, this would be extremely clear like glass, but since this eye has been preserved, the preservative has denatured the proteins and caused it to turn cloudy. Around that, we see a lot of periorbital fat and connective tissue. And this connective tissue and fat plays an important role because this eye is located within an orbit, a bony orbit. And if you turn your head quick or you got a blow to the head and you didn't have this periorbital fat and you did not have all this strong connective tissue, then the delicate structures of the eye would easily be damaged. Let's look at the posterior surface of the eye, and we see this long tube-like structure coming out the back of the eye. That is the optic nerve, and that will go straight to the brain. Some of the other structures that I want you to see here is just some of this brown material here. See a little bit of brown material over here, and you can see pieces of it here where it's been cut, and also over here a little bit. Those are eye muscles. Those allow the eye to actually move up, to move down, to move left, to move right. Now let's go ahead and begin by removing some of this periorbital fat. So I'm just going to take the scissors and I'm going to attempt to cut some of this fat and material off, sort of clean up the eye a little bit so that we can see things a little bit better. It's starting to look a little bit better. And that looks good there. We'll move some of this material out of the way. And you can see the optic nerve much better now, coming out the back of the eye. We'll talk a little bit more about that in just a few minutes. We want to start the internal dissection by cutting the eye right just posterior to the cornea. Now under some of this connective tissue is going to be another layer called the sclera. And we'll see that a little bit more clearly after we cut into the eye. So what I'm going to do here is I'm going to take a scalpel and I'm going to hold it a certain way because I don't want the scalpel to slip. And this sclera that I was talking about, this material, is very tough to cut through. So I'm going to take the scalpel and I'm going to hold it in the palm of my hand like this so that the handle cannot move backwards. And I'm going to put my hands around it, just like that. And I'm going to take the scalpel. Be careful to have your fingers on either side of the eye. And I'm going to put it just posterior to the cornea. Right around there. And I'm going to gently push straight down. You can see that the eye is, there's liquid inside the eye. And this liquid and jelly substance inside the eye helps maintain the shape of the cornea and maintain the inner shape of the eye. We'll take a look at that. When I cut through here, you're going to see some liquid actually come out. And that liquid is called aqueous humor. And that liquid, one of the things that liquid does is it helps maintain the shape of the eye. We certainly don't want wrinkles in the eye as light is passing through it. So let's go ahead. I'm going to push down gently, straight down, straight down. There we go. And you can see some liquid come out. You can see the liquid begin to come out. And all I'm going to do is roll the eye and gently cut. Gently cut while I'm rolling the eye. Be very careful of your fingers here. If you want, you can use a pair of scissors to also do this. I find it a little bit easier to use a scalpel. And be sure not to disturb the inner structures of the eye when you're doing this. And I'm going to open that up. I'm actually going to cut this right into two pieces. And let's take a look at the posterior aspect of the eye first, and then we'll look at the anterior part second. So I'm going to move this out of the way. And all the liquid that you're seeing right here, this is aqueous humor. The jelly-like material, that is vitreous humor. And it serves the same function. It serves to help maintain the shape of the eye. So we can see that there. What I'm going to do is I'm going to gently try to get rid of some of this vitreous humor, just kind of pour it out, nudge it out the side of the eye. And then I'm going to take a paper towel and I'm just going to dry this up just a little bit so that we can see clearly in here. And we can see this cream-colored material that coats the inside of the eye. This cream-colored material is the retina. This material is probably the most important part of the eye as it contains all the photoreceptors, the light detectors that actually detect the incoming light. Now as I move this material, as I move the retina, you can see how I'm bunching it up and I'm moving it. It's actually going to be stuck to the eye at a certain point, right here. We'll see this in just a minute. But one of the things I want to talk about here is what keeps this retina pressed against the inside of the eye. The vitreous humor actually does. The vitreous humor is going to press that retina against the inside of the eye. And when the vitreous humor is not present anymore, I can easily take this retina and I can just remove it and I can bunch it up. And you're going to see that when I bunch it up, it comes off rather easily, except in one place. In one place.
Look at the color underneath.
And we're looking, there it is. You can see that one place right there where it appears as though the retina is passing through a hole in the eye. What that is that's actually the axons of the nerves in the retina. All the axons of the nerves in the retina are passing through the back of the eye. And where they pass through right here, the spot in the retina where they actually pass through is referred to as the optic disc or blind spot because light cannot actually be detected there. Now all these axons pass through the back, and if we look exactly opposite of that optic disc, that is where the optic nerve, all those axons are going to make up the optic nerve. That is where they exit and go to the brain. Now going back to the eye again, that's the retina. The next part that we come upon is going to be this iridescent, metallic looking structure here. Now humans don't have this, but animals that are nocturnal, that are active at night, or animals that are corpuscular, active during twilight, early morning, late evening, that actually helps them see better at night. That actually reflects additional light and allows that additional light a second chance to strike the retina. Humans, however, do not have that. This structure is called the tapetum, Now, if I remove that, and actually the tapetum is going to be continuous with this other black layer you see here, that's called the choroid. The choroid is very vascular, contains a blood supply that serves the eye. The black color of this choroid also serves to absorb light. So when light comes into the eye, a lot of it is going to strike the retina and get absorbed, but extra light that passes between the nerves of the retina is actually going to strike this choroid and get absorbed, so it's not reflected around the inside of the eye and we don't have blurry vision. Under the choroid is going to be a thick white layer. Now it's stained a little bit black from the choroid, but I'm going to get the forceps in here and you can see how thick it is. I'm going to peel back a little bit of the connective tissue here and you can see this very thick layer. That thick layer is called the sclera. And you can see as I push in on it, it actually springs back to its original shape. Very, very tough material, and that's why we had to hold the scalpel a certain way when we began to cut the eye. Now let's move on to the anterior part of the eye. And again, I want to remove some of that excess vitreous humor. So I'm going to remove some of that very gently. Don't want to disturb the other structures of the eye. So very gently I'm going to remove some of that vitreous humor. And I'm going to take the paper towel and just very gently dab it a little bit so we can see clearly in there. You can see all that jelly-like material. Now, the first structure we notice is this gumball-shaped structure. This is actually the lens of the eye. This is the lens. It's responsible for, it actually changes shape, and it's responsible for focusing the light on the proper place in the back of the eye. Now, again, in a live cow, or you, this lens would be very clear, like glass. Now, this one's hard, but in a living organism, this lens would be very soft. You could actually squeeze it like you could a contact. Again, the preservative has denatured the proteins, made them cloudy, and made them very firm and rigid. Now let's take a look at this black structure that surrounds the lens. So you can see all these striations. That is called the ciliary body. And it is actually attached to the lens, and that ciliary body is going to be responsible for changing the shape of the lens. If we look close or if we look far away, that ciliary body is going to contract accordingly and change the shape, either flatten the lens out or make it more spherical like it is right now. I'm going to slowly remove the lens, and as I do, we're going to see if we can see where the ciliary muscle is actually attached to the lens. And you can see look, almost looks like saran wrap attached to the lens right there. You can see the attachment there. Those are suspensory ligaments, and they attach to the ciliary body, and that controls the shape of the lens. I'm going to slowly pull that out, and you can see those suspensory ligaments tearing in two. And I'm going to remove the lens. So I'm going to remove the lens. If we were to cut the lens, it would look essentially the same. Might look layered inside, a little bit like an onion. So I'm going to move that out of the way. And we're going to take a look at some of these additional structures in here. Just going to move some of that out of the way. And medial to the striated substance was the ciliary body, is going to be a more smoother looking black structure. That smoother looking black structure, and I'm going to take the forceps and I'm going to grab it, is called the iris. That is responsible for allowing more or less light to enter the eye. During the day, you want less light, and at night, you want more light. So that iris is actually going to change shape, and it's going to allow this hole here, called the pupil, to become larger or smaller. That hole in the iris is referred to as the pupil.
All right, folks. Any questions? Pretty cool, right? All right. So the next thing we're going to do now, we're going to do the in-lab activities. We're going to jump on the Ed puzzle with the email that I sent you earlier. And you're going to find 3 videos in there. Okay. You have one of the videos is a model of a human brain. The second video is a model of a human eye. The third one is a model of a human ear. They're all under 10 minutes, so they don't take too long. Make sure you watch them until the end, answer all the questions as you move along. And after you're done with the Ed puzzle, you're going to jump on Lolima. There are 2 quizzes today, not just one. There's a quiz for Lab 7 and a quiz for Lab 8. Make sure you complete both of them. After you're done with the quiz, you're good to leave. Okay? We good? All right, let's get to it.